DNA structure in function. Guys, we're going to continue here. So in interphase, remember that DNA is in the form of chromatin, okay? And chromatin forms from what? DNA and protein, okay? So these, when we combine histones with DNA, so 8 histone molecules with DNA wrapped around it, what do you call that?
Nucleosome.
Nucleosome. Don't forget that, okay? So nucleosomes.
DNA plus histone.
Yeah, and DNA is in the form of chromatin and interphase, okay? So in the form of chromosomes, it's in from prophase and onwards, okay? So in the form of, if I ask you what sort of, if it's duplicated in the form of chromosomes, would you say, which phase would you pick?
Duplicated and meiotic.
The mitotic phase, okay? Or you could say in prophase or metaphase or whatever, okay? Not in G2, not in S, not in G1, not in G0, okay? But if I say if it's in the form of chromatin, duplicated, is it in G1? Is it G1? No.
What?
Yeah, but it's not duplicated.
Oh, okay.
ما هو بالاكس سيروم يصيرون بس في البروفيز والميتافيز
وش الكروماتين؟
اي يصير باكس بيصيروا الثنتين يبعض هاللون.
Okay? So if it's duplicated, what in the form of chromatin, which phase will it be? In the form of chromatin. Yeah, which part of interphase? S and G2, okay? So usually this sort of chromatin we don't see, okay, under the microscope, but it's only can be seen the DNA when it forms chromosomes. Yes.
Chromatin is nucleosome with each other, right?
Yeah. So chromatin is from the nucleosomes. Which what are the nucleosomes? They're 8 histones, DNA wrapped around them. Okay. So chromatin in S and G2 is duplicated. Okay. But chromosome in the form of chromosome is in
Prophase.
from prophase, okay, or mitosis, okay, mitotic phase.
Chromatin immobilis.
So this is what we're saying, okay? When you got a single yes. Yes. 8 histone proteins with DNA wrapped around them, okay? I mentioned it, I skipped it because I already told you that, okay? It's written there, okay? Here. So chromosomes are only visible in the mitotic phase. Unduplicated, okay? We get S phase, replication, we get duplication, but we only see this in the prophase, and they're called sister chromatids attached to each other by the centromere. After the mitotic division, you separate it and form unduplicated again, 2 unduplicated. Now, so we'll get back, when I get to mitosis, I'll explain that, alright? I think it's just basically the same thing here. So sister chromatids are identical, okay, to each other.
Well, when separated, we don't call it chromatin.
Yeah, single chromosomes. This karyotype is very important to know, okay? With the karyotype, we can visualize chromosomes, okay? So when later on I talk about the chromosomal disorders, you can detect them with that. You because changes in chromosomes, alright? The karyotype can detect that, alright? But it can also show the chromosome number of an organism as well. For example, it can tell you whether it's male or female as well. It can tell you the different type of chromosomes. It has this unique banding, which is used to detect those abnormalities, okay? So deletions can change that banding. Changes like extra chromosomes can be seen. One chromosome missing can be seen with this karyotype, okay? So it's very important in studying in the field of what we call cytogenetics, okay? When they study chromosomes. Now, the other thing is that we said that chromosomes occur in homologous pairs. So you receive one from your mother and one from your father. So when you receive it from your mother, we call it paternal. And when you receive from your mother, we call it maternal, alright? What they have is the same genes, okay? But in slightly different versions of it. Remember, we call those genes alleles, okay? When we talk about genetics later on, alright? So the different alleles are on the homologous chromosomes. So the difference between diploid and haploid, okay? In mitosis, we start with diploid cell. We end up with diploid. In meiosis, we start with a diploid cell. We end up with haploid. What does haploid mean? Half the number. So if a cell started with 40 homologous pairs of chromosomes, okay? 40 homologous pairs, okay? After meiosis, how many chromosomes in each cell? 40, correct? Everybody agrees with that?
40 single.
40 single, huh? Not pairs.
20 pair.
If I say it started with 40 chromosomes, and then how many chromosomes after meiosis? 20. Okay? Because I said pairs, homologous pairs, 40 homologous pairs is actually 80 chromosomes, huh? Or 40 chromosomes, when I say, is 20 homologous pairs, okay? So diploid we write as 2n, okay? In humans, what's the diploid number? 26, huh? 26. What's the haploid number? Sorry, it's not 26, 23. 23. Who said 26? Somebody confused me.
46.
Did you say 46? Oh. So the haploid number is 23 single, huh? 23, which is n, okay? Or 23 to 46, okay? 2n.
23 homologous.
Where do you find the diploid cells? Somatic cells, okay?
Yeah.
What about haploid? Gametes, okay? Where are they made? Where are they made?
Testes and ovaries.
Testes and ovaries, or the gonads, okay? So make sure you know the difference between those two.
Doctor,
Yes.
the germ cells, some of the germ cells are haploid, or all of them?
Well, depending on so the gametes will be, but you know like spermatogonium and oogonium, they're not haploid, they're diploid. Okay. So the spermatogonium and the primary spermatocyte, or the oogonium and oocyte, are diploid.
Okay. After meiosis, they will be.
After meiosis, they will form haploid. Okay.
So in general, germ cells are diploid, right?
Well, the cells that give rise to the gametes, yes, they're diploid.
What is the somatic cell?
Somatic cells is any cell in your body, like muscle cells, fibroblasts, osteocytes, the neurons, the neuroglia, all these other cells, other than the gametes. Yeah. 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 although there is some things that are unique, like red blood cells, they actually have done the mature red blood cells. They don't have a nucleus, so I'm not sure if you classify them as diploid or not, because they have no DNA. Okay, so chromosome. How many chromosomes are homologous in the male? 22. How many are in the female? 23. Okay. So for all your chromosomes to be homologous, what do you have to have?
For female XX, for male XY.
No, for all your chromosomes to be homologous, what do you need to have?
The same
44.
You need to have
44 chromosomes.
One from mother, one from
22 autosomes. Okay, remember the autosomes and X plus X, huh?
Yes.
Not XY, huh? All your chromosomes will be homologous. Everybody got that?
X and X.
Is X and Y homologous? No. So a male will not have all his chromosomes homologous. A female will have all the homologous. Okay? So if you ask how can you have all your chromosomes to be homologous?
You have to be
22 autosomes, X, X. It's written here, right here. So, in terms of haploid, you find it in the gamete, so you have 22 autosomes, either X or Y. Okay? So, if it's the sperm, you can get X and Y. If it's the egg, you can only get X. Okay? Alright, the cell cycle. So, we'll talk about the cell cycle in terms of mitosis, okay? First. So, the two phases, we have the interphase, which is the,
the longest.
between the cell divisions. This is the longest. You get duplication of what? DNA. Duplication of? When is the cell the smallest? Beginning of G1. So these points, remember them all, okay?
Can you say that again?
What happens in the interphase? What do we get? Duplication of DNA,
And organelles.
organelle. When is the cell the smallest? When is this the largest? End of G2, okay? Mitotic phase is the actual division, okay? The nuclear division. And and cytoplasmic division is the cytokinesis, okay? So mitosis, in mitotic phase there's two different stages. Mitosis, which is the nuclear division, and then cytokinesis, which is the cytoplasmic division, okay? So interphase is the longest, okay? And we have three different subphases. G1, this is where the cell is the smallest, okay? Because it just came from division. It has, this is where you get duplication of the organelles, okay? And it's before the S-phase, DNA synthesis, okay? And this cell is the smallest in the beginning.
What do you mean by smallest?
Because it's just divided into two.
Okay.
It's the smallest, the size. So this cell surface to volume ratio will be the largest in this, okay? Because the smaller the cell, the bigger that surface to volume ratio is. Remember from chapter 3? You remember that?
Yep.
Surface to volume ratio increases as the cell gets smaller. Surface to volume ratio decreases as the cell gets bigger. Yeah. Okay, we got that?
Yes.
In the G0, some cells will remain in G0, okay?
Like nerves.
They will never come out of this again, okay? So in adolescents, like nerve cells or osteocytes, and some muscle cells.
Doctor, if someone breaks his bone, does the osteocytes still remain in G0?
Yeah, they have other cells. Osteocytes are actually derived from a cell called osteoblasts. So we have plenty of that. We're always making the bone, okay? So it doesn't matter,
Oh,
okay?
so the body.
Yeah, the body can replenish the osteocytes by this osteocyte will not go back into the cell cycle, but the stem cells that make it are there. But we don't. This one is a different case here.
But osteoclasts can go.
Osteoblasts differentiate into osteocytes,
What is the muscle cell will remain in G0?
like skeletal muscle. Once it fuses the cells, during embryonic development, skeletal muscle. The cells fuse together to form these large cells, multinucleated. They stay in that. They don't go back into the cell cycle. Okay? After adolescence means after puberty. Okay. Now let's go to the other. So in the S phase, pretty simple. The DNA is duplicated. In the G2, slow growth, but it's preparing now for cell division.
So it's now bigger.
Now it's the largest, okay? Biggest in size, but in terms of surface to volume ratio, it's smallest, lowest surface to volume ratio, okay? And the beginning of the condensation of DNA from chromatin into chromosomes, okay? But they're not visible yet. Once they become visible, we get into prophase, okay? So which we'll talk about now. So mitosis and cytokinesis.
In G2 they are visible, chromatin duplicate.
In G2, they're not visible.
Chromatin.
In prophase, they become visible.
It just
Invisible.
duplicated from G2.
Yeah.
After that will be
Chromosome. When they become visible, then you know that you're in prophase. Okay?
for the end of G2.
Prophase. End of G2, it starts to begin to condense. That's the start of it. Okay? Alright, so mitosis. Remember the four phases? Prophase. One thing to learn is prophase and telophase are the reverse of each other. Okay? So in prophase, we get the nuclear membrane disappearing, the nucleolus disappearing, the chromosomes forming into two sister chromatids attached to each other by the centromere, the centrioles, the spindle fibers are forming and migrate to the opposite poles. Don't forget that, okay? It's in the text there as well on the next page, I think. Do you want me to go through it?
No.
From here,
That's easy.
okay, so in mitosis, do you get identical cells or non-identical? How many? Two identical cells. Okay? Don't forget that. The difference between meiosis and mitosis. Mitosis, two identical cells. Meiosis, four non-identical cells. Okay? And we start with diploid, we end up with diploid.
You said that meiosis cells are not identical because they're crossing over.
Yeah, we will get to that when we get to that section, okay? Because it's important information there. I will tell you about it. Okay, so let's have a look quickly here. As I mentioned, in prophase, chromosomes are visible. Mitotic spindles form from the microtubules. The centrioles start to move to the opposite poles, opposite sides of the cell. Okay? The nuclear membrane disappears. The chromosomes start to appear. Okay? And here the metabolic activity is low because it's now concentrating on cell division rather than normal activity. Okay? It has the priority of dividing the cell. Metaphase is pretty easy. In mitosis, the metaphase, 46 chromosomes line up, okay?
Okay.
Duplicated chromosomes. And the what's helping to keep this, make sure you know this, okay? That the chromosomes are pulled to opposite direction by each force. That's what's keeping them in the center, alright? So when you see the chromosomes lined up like that, we know that they are in metaphase, okay? So 46 chromosomes lining up there. Then once they separate, the sister chromatids are separated, okay? They're no longer now duplicated, right?
Yeah.
They pull to the opposite side, and this is happening by requirement of ATP energy, alright? So, usually in anaphase, okay? Which, remember this, it's the shortest period, okay? And the other thing is that usually cytokinesis begins in anaphase, the beginning of it. Okay? Two important things to know. Alright?
What's the second?
It's the shortest phase, and also cytokinesis begins, and it requires energy, and the chromosomes are now unduplicated, okay? They separated.
The centrioles will decrease the tubulin, so this will happen.
Yeah.
Central.
They will pull, that's the pulling force. We don't need to know the details of that, okay? But important to know that this is the shortest phase and the cytokinesis begins here. In the telophase is the reverse of prophase, okay? It's pretty simple, okay? So the chromosomes decondense, the nuclear membrane starts forming again, the centrioles disappear, the mitotic spindles disappear, okay? The nucleolus appears again, and the chromosomes form chromatin, okay?
And the cleavage furrow appear.
Yeah, and this cleavage furrow will appear here, okay? Bulging, and that's caused by actin rims, okay?
Doctor, so chromosome at opposite poles, is it telophase or metaphase? The chromosome, when they're at opposite poles.
Well, in telophase.
Telophase?
Yeah, they're starting to pull towards the opposite sides in anaphase.
On anaphase.
Metaphase, all the chromosomes are lined up, haven't separated yet.
They're not opposite poles.
No. Only the centriole is in the opposite pole, not the chromosome.
Yeah, okay.
Doctor, metabolic activity decreases only in prophase.
Well, throughout this, more concentration is on the division, okay? So the cytokinesis, so we have the nuclear separation, we need to have the cytoplasmic, which is the cytokinesis, and this is caused by a ring of actin microfilaments. Remember the microfilaments? That form a ring around the middle of the cell, the equator, and this ring starts getting smaller and smaller and smaller until it pinches off the two, and it's on the inside of the cell, okay?
Need ATP.
Yeah, this is energy, you know. Without it, we can't, you know. So what happens is it causes a constriction in the middle of the cell, and eventually it separates the two cells, and you get two new daughter cells. All right. Then after that, the microfilaments will disassemble. Okay. From the inside, it's pulling. Okay. But that detail is not important for you. Okay. What you need to know is the microfilaments are forming the cleavage furrow, and that's what's separating the cytoplasm, okay? So in the end, you have the identical diploid cells. You start with diploid cells, in the end, you have identical diploid cells, okay? So in mitosis is identical to diploid cells. Huh? So, this one is very important. So, what's happening with cytokinesis is
Separate.
This contractile ring is causing the contracting and pinching the two cells. Okay? It's made on the microphone. Okay, so meiosis. We find a lot of similarities between mitosis, but we will talk about the differences, okay? Now in meiosis, how many cells are produced? 4 non-identical. Haploid. We start with diploid. When do we get haploid? In which? Meiosis 1 or meiosis 2? Meiosis 1. Yes. Mine that meiosis 1 is haploid. We're separating homologous chromosomes, remember?
Yeah.
So what do we get? At the end of it is haploid.
But in the form of the duplicated chromosome.
In duplicate. Yeah. The second step is to separate the sister chromatids, okay?
So minus 1 haploid, minus 2.
Also haploid. Once it's haploid, okay? Okay, so let's quickly move on.
Doctor, meiosis 2, we start with haploid.
Yes. Meiosis 2, we start with haploid cells.
Minus 1 we start with.
End with haploid.
Yeah. So that's why it's called a reduction division, because it reduces the number of chromosomes, okay? The word reduction division is talking about getting haploid from a diploid cell, because reducing the number of chromosomes. And that's for which process is that important for? Why is that important to reduce it? Yeah. For reproduction, huh? For fertilization. Remember that, okay? To get haploid cells is important for fertilization. That's why it's important, okay? Because once we get fertilization, we are returning back to diploid, alright? So this meiosis is important for fertilization, okay? That we reduce the number of chromosomes. Okay? So several things happen in meiosis. One of them is that to get that genetic variability, okay? We're getting crossing over, so this mixing and reshuffling. Mix and reshuffle. So remember independent assortment of the chromosomes. Law of independent assortment in the next chapter. This is what happens here, reshuffling, and also mixing of the genes by crossing over, and we're getting half the number of chromosomes. Okay. So in the end, the donor cells are haploid, and they're genetically different to the original cell. All right.
Haploid and
Non-identical.
Because of crossing over.
Yes.
I'm not sure. Just maybe please explain the part with the fertilization.
So what function is this important for? Meiosis? It's important for fertilization. Okay? That's the important because once you get half fertilization, you restore diploid. Okay? Yeah. So that's it. Yeah.
Doctor, what's the meaning of fresh food?
Huh?
Fresh food. Fresh food.
Which the last slide?
Before.
Next,
Reshuffle. Huh? You know, you got some cards. You're shuffling them, mixing the cards. You know, shuffle. You know the chromosomes are reshuffled. Okay. I'll show you what happens. Okay. Chromosome 1. Okay. Chromosome 2. All right. They come in homologous pairs. All right. So you got one from your mother, one from your father. Okay. It lines up here, and the father one lines up there. Okay. On the next chromosome 2, on this side, your father's one lines up, and on this side, the one that you got from your mother lines up in meiosis. So this can happen for all 23 homologous chromosomes, and it's happening in meiosis 1. Okay. This is a reshuffling. Okay. the law of independent assortment. So this pair doesn't depend on that how it assorts. This one can go this way or this way, this way, this way. And we say we get 2 to the power of 23, was it? Correct? Which is 8 million, huh?
Yeah.
8 million different combinations.
Also for the
More than 8 million. Yeah, this is the reshuffling, okay? So before we enter meiosis, we need to duplicate the DNA, okay? So the DNA is duplicated. You get an S phase, duplication of the DNA. So cells that are the what type of cells? What type of two cells
Identical.
we get that give rise to it? The primary spermatocyte and the primary oocyte. They both have
Diploid.
duplicated chromosomes and they diploid. Diploid, okay? So this the precursor is referring to those two types of cells, alright? These precursor cells are so the spermatogonium undergoes duplicates the DNA, okay? And forms the primary spermatocyte and primary oocyte, okay? So these precursor cells means the cells before we form the gametes. What are they formed from? Primary spermatocyte or primary oocyte. These cells are, they have duplicated DNA, okay?
So the result of meiosis 1 is diploid cells. 2
Meiosis 1,
No.
haploid.
haploid cells.
Begins with the
I'll explain it to you in a minute, okay? You will understand it. So it's 2 divisions, meiosis 1, meiosis 2, and each one is numbered according to the phase according to the meiosis. Whereas meiosis 1 is going to be anaphase 1, meiosis 2, anaphase 2, okay? Like that. So let's have a look at meiosis 1. Typical prophase, okay? With some differences, okay? The chromosomes condense to form, the chromatin condenses into chromosomes. Spindle fibers forming, centrioles move to the opposite side, nuclear membrane disappears. This is where we get the difference, the crossing over. So remember, exchange of segments of DNA of the chromosomes between non-sister chromatids, okay? If we have exchange between sister chromatids, what will happen?
Variation.
Okay? Because they're identical, it's a copy of each other. So the crossing over must happen with the homologous pair, okay? Non-sister. We got that? So that is crossing over. So you see now you got a new variety of chromosomes that don't look the same as the original ones, yeah. Okay? This is why we get the genetic variation, 1-4. Crossing over is happening. So, typical metaphase lining of chromosomes except homologous chromosomes line up, okay? You see that?
Yes. Double line.
So the one from the mother, say, and one from father that you got, line up, okay? Homologous pairs. So how many homologous pairs will line up? 23 homologous pairs, okay? So 23 homologous pairs, 23 pairs. So you got the 23 from your mother and 23 from your father. So they're pairs, okay? So when you separate them, you end up with 23 chromosomes in each of the new cells. That's why it's haploid, okay? That's why it's called reduction division. Because what you did is, you reduced the number of chromosomes to half. So, the major difference between mitosis and meiosis 1 is in metaphase, I'm talking about. Sister chromatids separate in mitosis, homologous chromosomes separate, okay? So, that's what's happening here. They're lining up. So, in anaphase, you get the separation of the homologous chromosomes. Are sister chromatids intact? Yes.
Yes.
Sister chromatids haven't separated yet. Okay?
And the crossing over.
This will happen in the next.
yeah.
Okay? Sister chromatids are still intact.
This is two.
So we have typical telophase, cytokinesis, like mitosis, the same thing, okay? The chromosomes arrive to the opposite poles, centrioles, the spindle fiber disappears, chromosomes decondense, nuclear membrane appears, you get two haploid daughter cells which are non-identical, okay? But the chromosomes are still in the duplicated form, okay? And there has to be another round of division. We don't have interface between them, okay? There's no interface between myosome. What do we call that between it? It's called interkinesis, okay? Remember we did in the lab? We had this? It was in the lab manual.
Yes.
So there is no interface. The cell goes straight into meiosis 2.
Is the nuclear envelope surrounding chromosome also here?
Yes. So everything will disappear and then it'll start again the next phase. The next round of division. So the nuclear membrane will appear again. It's the same as the prophase, okay? Okay, meiosis 2. So we understand in meiosis 1, homologous pairs separate. Crossing over happens. Also we're getting some reshuffling of the chromosomes as well, as I said like that. Meiosis 2 is similar to mitosis, exactly, except less chromosomes, 23 chromosomes, okay? So instead of 23 pairs, we got 23 duplicated chromosomes, okay? So remember these differences. So here we have, these are non-homologous pairs, okay? So you get 23 duplicated chromosomes lining up, okay? So the sister chromatids separate, okay? Sister chromatids.
So
Okay.
this two is just completely like mitosis.
Yes, except 23 sister chromatids, not 40,
46.
46, okay? 23, not pairs. Mitosis 46 chromosomes, not pairs, okay? 46 will line up. Huh? 2.
And between the first cytokinesis and the second group phase is interkinesis.
Yeah. The final result is that now we have 4 haploid cells, okay, with 23 chromosomes that are non-identical, okay?
Because the crossing over.
Crossing over, reshuffling, and so on. All right?
Doctor,
Yes.
can you come back? In metaphase 2, 23 pairs or no?
It's not pairs, 23 duplicated. Like mitosis. This is what's happening in meiosis, okay? One. You're getting like this separation, okay? Once that's separated, now each cell has, what does it have? It has duplicated chromosomes, not pairs anymore, okay? So in meiosis 2, we separate those two, okay? You end up with a cell with single chromosome. Unduplicated. You understand that?
Yeah.
Yes. 23 duplicated in terms of the human chromosome when we're talking here.
What interkinesis occurs?
Huh?
Which phase? At which phase? Interkinesis.
Interkinesis between the meiosis 1 and 2.
So we don't say cytokinesis. We say
No,
interkinesis then
interkinesis,
minus.
yeah.
Okay. So when is cytokinesis?
Sorry, doctor, what he's talking about?
Now, there is cytokinesis. There is no interphase.
Yeah.
There's no interface. There's no interface between them. It's called
So the first cycling is called interference.
no.
So between cycling first and prophase 2.
You know how after the cells are divided into two,
Okay.
we don't go into interphase again. You know, there's not a long waiting period or anything. There's no DNA duplication. We already done it.
Yeah.
Okay? So it's duplicated. So straight into the second division. But the period we just call it, a name is called interkinesis between the two meiosis. There's no interface, okay? We got that? Is that clear, everybody? Yes. Yes. This chapter is nearly finished, okay? It's still 2:30. Salat is not done yet. We'll take a break after we finish this chapter and we'll come back, okay? Because there's only a few sections of not much. Is it? Yes. Okay. Alright, let's get on to gametogenesis. So basically, we're talking about the formation of the gametes. So when we're looking at in the sperm, we get 4 sperm compared to 1 ova, 1 egg, okay, functional. So in the males, the process is known as spermatogenesis. We start with this cell called spermatogonium, which is diploid. So it goes mitosis and growth. It has duplicated chromosomes, okay, because it went through S phase,
This we call
and it forms the primary spermatocyte, okay?
We call it germ cell, diploid.
Yes, yeah, these are the germ cells. So, in this stage here, it's diploid, okay? These cells. Okay? That, any cells this side are haploid, alright?
But mitosis doesn't happen to.
Now, to give rise to the primary spermatocyte, okay? From this cell. Okay? This cell gives rise to that. And then that undergoes meiosis, okay? So, we get the primary spermatocyte, which is diploid. This undergoes meiosis 1. We get secondary spermatocyte, which is haploid. And then we get meiosis 2. We get the spermatids, still haploid. And then they mature, okay? To form the sperm cells, which are haploid, alright? Basically, just need to know the names. And what the structure of the chromosome is haploid or diploid? Yes?
Can you repeat what's the difference between spermatogenesis and primary spermatocyte?
Well, this is diploid and that's diploid. So it undergoes duplication and growth, okay? To form this. You see now the chromosomes are duplicated. This is the cell that will undergo meiosis, not that one.
So it's not the same. with
This one
bigger.
forms these cells, okay?
After interface.
After growth.
Yes.
Bigger and not.
Yeah, and the cells that are formed, the primary spermatocytes are formed from the spermatogonium, okay?
So not the same. but
Yeah,
bigger.
so that's why we have, unlimited supply of the sperm, because these cells are always mating.
Oh.
But in terms of the eggs, these run out. They're already established. Female has like all the and the
Fix
form
them
of
up.
the
Fix them up.
the primary the primary oocytes. Okay. Yes.
Can you repeat what you said, please?
So the spermatogonium is like the cell that gives rise to the primary spermatocytes. Okay. So in the males we have these cells are always making these. That's why you have a continuous supply of the sperm. In the female, all the cells are in this.
Primary outside.
Yeah.
Because of that,
They only have like a limited supply, 400,000. Okay.
But
They release 1 egg every.
But it goes under growth, not mitosis.
Yeah. To form this, you need to divide this cell.
Also, it will.
Divided.
We will have 2 cells.
Yeah.
Two
Well,
cells. will
2 cells, whatever,
Yeah.
20 cells, a million cells, 5 million cells, 300 million cells.
Meiosis,
Yeah.
meiosis 2.
Yeah.
Meiosis
So
Wow.
this is the one that will go meiosis 1 and 2, not this one.
The first
This one
will go,
forms those cells.
the first one will go just mitosis.
Yeah.
Mitosis will go.
Yeah.
So spermatogenium
Spermatogonium is the cell that gives rise to the primary spermatozoa.
gives two
Keeps
cells.
making these cells.
Two cells, right?
Yeah.
Primary and spermatogenesis.
They're always replenishing the supply of primary spermatocytes. If you look at the semiluminous tube, remember the tube? You can see the different stages of these cells in the laboratory. If you remember the histology of it.
This slide, what's important?
The important is to understand the cell of the spermatogenesis, what the status of the chromosomes are. Here it's diploid, here it's haploid. Okay? This one is easy. When you go to look at the oogenesis, it's just slight difference in the formation of these polar bodies. Okay? So the same thing as we saw for that, the oogonium gives rise to the primary oocyte, and this is the cell that will undergo meiosis 1 and 2. But the difference is that in meiosis 1 in oogenesis, one functional secondary oocyte is produced, which is haploid, and the one polar body is produced. Okay? The function of the polar body is to remove the excess chromosomes. Okay? The 23 duplicated chromosomes. It acts like a trash, a junk, to get rid of the chromosomes. That's why in meiosis 1 we get unequal cytokinesis. Okay? Because only a small portion of the cytoplasm ends up in the first polar body, the rest ends up in the secondary oocyte. The reason for this is so we have lots of nutrients and supply when the egg is fertilized, so it can divide in the first stages before it's attaching to the mother's womb. It's got a lot of energy, a lot of nutrients, lots of organelles, right? In the initial stages when it's fertilized. That's why all of the cytoplasm ends up in the secondary oocyte. And only a small portion of that is removed with the polar body. Okay?
So
Yes.
the primary oocyte doesn't insert meiosis 1 until it's fertilized, right?
No, meiosis doesn't complete meiosis 2.
Doesn't complete meiosis 2
Yeah,
until it's
yeah.
fertilized.
But we don't need to know that, okay? We don't need to know that. It doesn't complete meiosis 2. So this chromosomes, it doesn't complete metaphase 2, okay? Yes.
So small body remove the
Unwanted chromosome, because we need to end up with haploid, okay? So this secondary oocyte then undergoes meiosis 2. It forms another polar body. Again, uneven cytokinesis, unequal. So only a small portion of the cytoplasm is in the first polar body, second polar body, and the rest forms the ovum, the egg, okay? The egg is haploid. So by the end of oogenesis, we can end up with 2 and sometimes rarely with 3 polar bodies and 1 functional egg which is haploid, and it contains most of the cytoplasm, okay?
So we mostly end up with 2 ovums.
No, 1 ovum. 2 to 3 polar bodies, okay? Sometimes this first polar body can undergo meiosis 2, okay? In the end, you will end up with 3, okay? But that is very rare, okay?
doesn't have the energy.
Yeah, rarely. That's why it says rarely divide. So 2 to 3 polar bodies and 1 functional ovum, containing most of the cytoplasm. Yes. Huh? No, 1 egg. Okay, that's what I'm saying. 1 egg and up to 3 non-functional polar bodies. Those polar bodies get destroyed and disintegrate. Yes.
When does ovulation appear?
So every month, ovulation is the process of the release of the egg. Okay. Now the secondary oocyte. Depends on the hormones, okay? The estrogen, progesterone, okay? In the female. So after the period, you know, when the female has the period, the next cycle begins. This is where the beginning of it
Every
is.
28 days.
But the egg is most fertile at around halfway through the cycle, at day 14, okay? This is when it's released. So ovulation is, I think, it's happening. Ovulation is happening in 13, 14 or something like that day after the beginning of the period. So when the period starts, the ovulation is so about 14 days, 13 days after that is ovulation, okay? But don't worry, we don't need to know those information because we've got a lot to cover. Keep the questions on the topic, please. We're going to be here till any short time if we don't finish.
What's to do with the
Huh?
What's the point?
The same thing as before. The different stages of the cells. The polar bodies are non-functional and they're formed by unequal cytokinesis. Their function is only to carry the waste chromosomes and they disintegrate. They get destroyed and the ovum is the one that is functional, okay? That can be fertilized. The reason that happens is so it has lots of energy, nutrients and organelles in the first stages of after fertilization. So the cell, the ovum divides rapidly after that. So so the reason is that you get unequal is so the egg gets, you know, all right, and the polar bodies, they remove the discarded chromosomes and they just disintegrate, okay? Yes.
Can you repeat why there is unequal cytokinesis?
Unequal cytokinesis because we want the egg, if it's fertilized, it has lots of energy to grow and survive and develop at the first stages, okay?
So in order to get that energy,
Yes.
it needs
Because the egg is fertilized before it reaches and attaches, okay? It needs energy from somewhere. Where's it getting it? It's not going to get it anywhere. Well, it has its own supply because of that unequal difference. The large cell, most of the cytoplasm energy, so it's fertilized, it starts multiplying, okay? As soon as it's fertilized, mitosis takes over, okay? The cell divides, and it needs energy to divide. And the first beginning, I'm not sure what stage it is before it attaches. Until it can get the energy from the mother, it needs to have its own supply, okay? And that's why unequal cytokinesis is important. How many slides do we have there?
Maybe 13, 20.
That's 32.
Alright, guys, I'll go quickly through here, okay? We know what a gene is, functional use of the DNA, okay? We have up to 20,000 different genes. So short segments of DNA, okay? Up to 20,000 of them, and they can give rise to proteins, okay? But not all the time, okay? They can have other functional RNAs, okay, like ribosomal RNA, tRNA, and so on. In terms of gene expression, we have two functions. Transcription and translation is very important for that, okay? So we'll cover that when we get to it, alright? So the functions in DNA are replication, transcription, translation, okay? In DNA replication, which is happening in the S phase, okay? Remember that the DNA is a double helix?
Yeah.
And there's complementary base pairing between them.
Yeah.
So ATCG base pairing, 2 strands. Before the DNA is replicated, we need to separate those two strands, okay? So there's enzymes that separate the two strands. Well, it's not mentioned in your book anywhere, but it's called helicase, okay? It's an enzyme that actually separates the two. You don't need to know that, but that's the enzyme that does it. So it separates the two, and then... Huh? It's in the book? Okay.
Doctor, what's the function of the DNA polymer?
I will get to that soon. So the DNA, the helicase separates the two strands, okay? We need to separate the two strands. And these two strands will act as templates, okay? As a template, so the cell can make another copy exactly to the original, alright? So if we have, for example, this is the double-stranded okay, DNA, we separate it. Let me write it here, it's better. So if we have the two, you can see it here anyways. You see you got G, C, I, T, C, G, okay?
Yeah.
You separate the two. So we use the same base pair, complementary base pairing to make the second strand again. So if we do that to both strands, we end up with a copy identical to the original one, two copies. Now how does that work? Once you separate the two strands, the free nucleotides inside the nucleus come and base pair with the, okay? So you have nucleotides in the nucleus, they will come and start base pairing with this. And then an enzyme called DNA polymerase, okay? DNA polymerase will join the nucleotides one to the other, okay? Now this is the replicating enzyme, alright? This is very important for DNA replication. If something goes wrong with this enzyme, you can't get DNA replication, okay? The cell's gonna die, alright? So, in some drugs in cancer, they target DNA replication because cancer cells replicate very fast. To slow them down, we use drugs against DNA polymerase, okay? So, if a drug targets DNA polymerase, it can stop division, cell division, okay? So we understand how DNA replication is working?
Stop the division cell.
If DNA polymerase is damaged or inhibited, cell division will stop. Important to know that. Sometimes also what happens is that we can get changes in the sequence, okay? Which are known as mutations, alright? These can happen by physical or chemical forces. Like in terms of physical, we have the UV, X-rays, gamma rays, and all sorts of these radiations can cause changes in the DNA sequence. Or you can have chemicals like hydrogen peroxide, some mutagens in the smoke, cigarettes, they have mutagens, chemicals. A lot of, in the past they used some pesticides, to kill the bugs. Some of them they actually cause mutations in the DNA. They don't use them anymore. So chemicals. Now, if you have mutations in the gamete, will that be passed on to the new generation?
No.
The gamete? The gamete. Yes.
Gamete, yes.
Yes. In the somatic cells?
No.
No. So, diseases that are caused by these in the somatic cells, like cancer, okay? They cannot be passed on to the offspring, okay? But if they cause in the gametes, like sickle cell anemia, or some of the traits that you have, like thalassemia, and so on, they can be passed on to the next generation because they happen in the gametes. How do we fix these problems? Well, the cell can has some mechanisms to repair. So the repair enzymes, the checkpoints, okay? We're not learning about the checkpoints, but just briefly about the repair mechanisms. So if a cell detects that in the cell cycle it has some sort of a mistake in the base pair that is added, because remember, we have complementary base pairs. So if one of the base pairs is wrong, it can use the template to correct it, okay? So for example, if here by accident it puts C, the there's proofreading enzymes, okay? Enzymes that detect errors, okay? It'll come and read it and it'll say, oh, this is the wrong. Then enzymes will cut this out, okay?
And replace it.
Replace it with the correct one, and then connect it back, okay? So these are repair enzymes. We have them in our cells because mistakes happen all the time. You know, you have replication happening all the time, you know, and mistakes happen, the cell recognizes them. But sometimes, unfortunately, it's not 100%, you know, there. And that's why we get diseases like cancer and other, you know, diseases that are caused, that are genetic, okay?
So there's a percentage about, you know, contest that when you get a cancer or the mutation about this.
Yeah, well, depending on the gene that is mutated, okay? So, not every gene will cause cancer if it's mutated, okay? Specific, like genes that are important in the cell cycle itself, control the cell cycle. If there's a mutation in there, it can cause cancer because the cell cannot check itself. It just keeps going and dividing. That's basically what it's doing in cancer. Cell cycle, or the cell has mechanisms. If there's some problem with it, it can cause cell death. If you have problems in the cell, in the genes that are checking for correction and then causing a cell to die, and that cell is surviving, not dying. So it's depending on the gene and the mutation, okay? How severe the mutation is, okay? For a lot of these cancers, we have mapped, you know, common mutations that will cause that particular cancer, okay? They can be used to detect it. Yes?
The enzyme that recognize the damage or the error.
It's
It's
DNA.
the same as the enzyme that inserts the correct.
Yes,
It's
it's DNA polymerase. DNA polymerase has actual proofreading. It can back up and read.
Yeah.
It has proofreading. It can go back along that strand it just created and read it and say to check if it's correct.
And the enzyme that reconnects is different from the first enzyme.
So there will be an enzyme. There's a set of enzymes. We don't know exactly what they are. Okay.
Yeah.
All right. So just for your level, we just say enzymes that repair these mistakes. Okay. And make sure you know this when they're active. It's important. Okay. So they're active between DNA replication, which is S phase, into the beginning of mitosis, which is the end of G2. All right.
It's in G2, right?
Yeah.
From S to G2.
All right. I don't need to go through this one. This is just a case study. So in transcription. Oh, sorry? No, that's just activity. To learn something about mutations, okay? We'll just go for it. We'll just finish this section and then we'll go for Salah.
Yes.
How does that sound? Still, Jama'ah is not done in the masjid. What time is Jama'ah in the masjid? 2:50. 3 or 5? 2:50 or 3 or 5? Okay, so it's already over. Anyways, we'll talk for 7 minutes. Okay, so transcription is the second function of DNA, converting the DNA code into the RNA code which the cell can use for its various functions, okay? Now, yes.
Is the DNA replication involved in protein synthesis or
No.
no?
It's one of the functions of DNA, okay?
Yeah.
The 3 functions that we listed. Converting that DNA message into messenger RNA, which is useful for the cell, okay? That's transcription. And remember that the difference between DNA and RNA? So the sequence is important to know the sequence, okay? So instead of thymine, we use uracil, okay? So it's a ribose sugar as well, not a deoxyribose, okay? So when you're writing the code for RNA, there's no T in it. When you're converting back from RNA to DNA, you must say, you convert U to T, okay? So the transcription is involved in transcribing the single unit, remember the genes, alright? From DNA to RNA, okay? Where does transcription happen? In the nucleus or the cytoplasm? Nucleus, right?
Yes.
So transcription is happening in the nucleus, and then that translation will happen in cytoplasm, okay?
The transcription happens in the nucleolus or in the
No, the nucleus. Nucleolus is where the ribosomal subunits are made.
The translation and RNA is protein.
In the ribosome translation. Okay? Important to note that the promoter is the start of transcription. Okay? This is the sequence that marks the beginning of transcription. Okay? So promoter. And transcription happens until you see the terminator which stops transcription, okay? Now the enzyme that is involved in that is called RNA polymerase. Remember in DNA replication it was DNA polymerase and RNA polymerase. So RNA polymerase attaches to the promoter, and then RNA polymerase is important in unwinding that DNA, okay? So it does it for only one. It doesn't. Remember that in replication, the whole of the DNA must be unwound. In transcription, only for one gene. Okay. Okay. So make sure you know that. So it unwinds it, and then remember that RNA is complementary. Also, we can get except for the U to T. So if you had a say we had this sequence. So I'll write it as double stranded, okay? So it'll be, alright, that's the DNA sequence, okay? The RNA polymerase binds it and separates those two, okay? So when it separates it, so once it binds, separates it, so then it will start adding complementary base pairing, not to this sequence, okay? It will use that because this is the gene sequence, okay? That's the sequence of the gene. So it goes from this direction, okay? To make this sequence in RNA form, it has to use this as a template, okay? Okay? So what will it add when it finds a T? A.
Oh. T.
U. Everybody got that?
Yes.
The complementary base pair. T, I. If it's I, because there's no T, we have to use U. Okay?
Because
This is RNA. Okay? If it's got C, we use G. If it's G, C, T, I. The only case we use U is if there's an I in the DNA, okay? Because the complementary is T, and T is not used in RNA, so we have to use U. Is that clear, everybody? Make sure you know that, okay? Yes.
It's a move from 3 to 5 or 5 to 3?
5 to 3 prime.
From
Yeah.
5 to 3?
5 to 3.
Not 3 to 5?
No.
What's the relation of transcription?
5 prime to 3 prime. That's the 5 prime. The DNA is 3 prime to 5 prime. Okay? You don't need to know that, alright? It's to do with the direction of how it's made. 5. You remember the phosphate in the hydroxyl group? Hydroxyl group is on the 3rd carbon, I think, and the phosphate group is on the 5th carbon. So hydroxy and phosphate bond, they bond together, okay, to form the phosphodiester bond, the bonding between nucleotides, okay, and they form it between 5 and 3 like that, okay. So if you have a sugar, like that, then you have the OH on the 3rd carbon, and I think there's a phosphate is on the 5th carbon, I think. Alright? Phosphate is PO3 or PO4? PO3. Okay? So the next nucleotide, okay, to join it, so we have to join another nucleotide to this, okay? So the next nucleotide, it will have its OH group here, whatever the structure, and phosphate PO3, okay? So, 3, 5. So, this is 5, sorry. So 3 and 5, okay? Carbon. What is that join? 5 to 3, 3rd carbon, huh? You got that? Yes, sir. Then again 5 to 3, 5 to 3. That's why it's called 5 prime to 3 prime. The synthesis of the RNA molecule is in this fashion. But this is beyond our topic, okay? The students are asking these questions, so we need to answer them. Alright. Our topic is simple like that, okay? You will learn that later on in biochemistry. Okay? So these 2 points are very important. Number 1, the promoter marks the beginning. Number 2, that the RNA polymerase unwinds a single gene, not the whole. Okay? So by the end of transcription, we end up with what we call a primary RNA transcript. Remember that, okay? Or RNA primary transcript, it says here. So the primary RNA transcript or the RNA primary transcript as referred to in your book.
I think it's called the pre-mRNA.
No. Primary RNA transcript, not pre-mRNA. Remember that the RNA, this is very important to understand this concept, okay? So have your attention here. The primary RNA transcript has introns and exons, okay? So these need to be processed before this can be translated, alright? So what happens? The introns are removed and exons are joined together. So make sure you know that, alright? So if you had a sequence, what would be the final? So if to remove, this is the primary transcript, okay? To get the final transcript, we need to remove these. That's the final, okay? Alright?
So remove the introns and then join the extrons.
Yeah. So if you're given that sequence to identify the final, that's it. Okay? You got that?
Yeah.
And the function of the introns is, so exons code for the protein, while introns don't, okay? But sometimes the introns can be spliced. Some introns can form part of the exons, so you can get a slightly different version of the protein, okay? So the introns can help in making a different version of the same protein. It's mentioned here. So the same gene can give a slightly different mRNA molecule. Okay? Now, the genetic code. How does the ribosomes read these mRNA? Okay? Remember the genetic code. We have the triplicate codon. So every 3 nucleotides of the RNA code for one amino acid. Okay? If we have a look at the genetic code, we find that there are 20 amino acids. Okay?
So what's the difference between genetic code and codon?
So the genetic code is the table here that identifies each codon, okay? Based on the corresponding amino acid. This is the genetic code, okay? A codon is referring to this one triplicate. No, we will give you this table in the exam. Okay?
So the genetic code is
This is the genetic code, okay?
of codes.
Yeah. The code, you know? How do we tell which amino acid? It's we have. This is the code. A codon is referring to a triplicate that correspond to a particular amino acid. Okay. Now, if you have a look at this code table, you can easily find out the amino acid. If you, the first letter of the triplicate, for example, you know it. If it's U, C, or I, or G, if it's U, you'll find it here. Okay, the first letter. It won't be anywhere else. Then what's the second letter? If it's U, C, I, or G, say it's C. So it's all the amino acid will be found here. Then you know which is the third letter. You can easily identify which amino acid it is. Okay, based on the first, second, and third letter. When we look at the code, we find that there is only one start. Okay, the IUG, and it codes for methionine. Okay, and we find that there's 3 non-coding. They don't code for any amino acids. Okay, we call them the stop codons. Okay, 3 non-coding. The rest of them, they code for the 20 amino acids. Now, if we have a look at the number of codons, we find that there are 64 different codons. But we have only 20 amino acids. Now, how does this cell get around this problem? It uses multiple codons from the same amino acid, okay? So, for example, serine here has 6 different codons, okay? Now, if I give you a sequence like this, the RNA sequence could be multiple different, okay, variations, okay? So make sure you know that, all right? For example, it could be UCU. Tyrosine, where is it? Up here. UIU. That's one variation, okay? Or it could be UCC, okay? And UIU, okay? The same thing. Then it can change. So you see that the messenger RNA could be many different variations, okay? But it will still code for the same amino acid, okay? Because we can use any one of these codons in the messenger RNA to code for the same amino acid. Do you get the point?
Yes.
Everybody got that point? Yes.
How many amino acids that are consist of one
Codon.
codon? Yes.
There's methionine.
CRP.
I think that's the only one. No, this one. Is that it?
No.
Yes.
It's the only one.
Yep. Tryptophan. Tryptophan and methionine.
So the thing is only made by AUG.
AUG. And it's the start codon as well.
Could we have it in the middle of the sequence?
Yes. Yep.
Do I have to memorize each amino acid.
I'm gonna give you the table in the exam.
Doctor, both tables or
You just need this one. Don't worry about that one.
we don't need names.
No, just need the letters. Okay?
How person ask me what's give me a chromosomal
I'll give you a sequence,
and what is it?
DNA sequence converted to RNA, or amino acid sequence converted to.
And I will give you the
If I give you RNA sequence converted to amino acid, okay? If I give you a amino acid sequence converted to RNA sequence, okay? Yeah, give me the codons.
You will give us start codon and end codon.
Whatever, you know, it doesn't matter. You can do it if you have the table. If you have the table, you can solve any sequence, okay? Because this is the code. As long as you have the table, you can solve any sequence I give you. You got that?
Finish.
Alright. Translation.
Happening in the cytoplasm.
Huh?
Happening in the cytoplasm.
It happens in the cytoplasm, but let's take a 10-minute break for Salah, okay? 10-minute break. Is that enough? Because I don't